Hello, grade 8 students. Welcome to our lesson today on food test. During this lesson, we want to achieve certain objectives. These are the objectives we want to achieve during today's lesson. During the use of our online simulation, you should be able to identify which test turns out to be positive or negative for a particular nutrient. And also, during our group and our class discussions, you should also be able to describe or explain to me which food could be present in various samples that has been used in your simulation or that has been tested in our simulation. First things first, do you know? So there are certain things that you have to know in order for us to start learning about food tests. So let's put on our thinking caps and think. What are nutrients? That is the first question. If you so desire, you can write that down in your notebook. Or if you can remember, that's fine as well. What are nutrients? I would like you guys to share your responses in the chat if that's possible for you. Second question, what are the major types of nutrients that you can get from food? We call them macronutrients. What are they? Final question, how are these nutrients used by your body? Let's see those responses coming in. Okay, so now that you have answered the questions, let us dive right into the answers of these questions here. What is a nutrient? Now, if you had said that a nutrient is a substance found in food that an organism needs to live and grow, then you are perfectly correct. If you had listed the carbohydrates, proteins, fats as the main nutrients that are found in food, then you are also correct. These are known as macronutrients. Now, of course, you also have vitamins and minerals, of course, needed in more smaller portions. Those are called the macronutrients. But we are basically going to be focusing on these three nutrients today. How are the nutrients used by your body? Of course, you have to eat the food, and through the process of digestion, the nutrients are absorbed by the body to be used for its varying functions. Now, just a little synopsis of all of this and eating that you are going to be doing during your entire lifetime. Do you actually know what carbohydrates, proteins, and lipids are, and some foods that you would get these nutrients from? Well, carbohydrates are a major source of energy for us. It is found in anything from sweets, fruits, potatoes, cereal, pasta, flour, and also other plant products. Proteins are used in body structures such as muscles, skin, and hair to help them grow. The rich sources of protein will include meat, dairy products, and of course, beans. Lipids, also known as fats and oils, are used for energy, insulation, and as an essential building block of cells. Meats, dairy products, and oily plants such as olives are very rich in lipids. Since recently, there has been a lot of discussion. I don't know how many of you have eaten the ice cream, the drumstick. There has been a lot of discussion on what the drumstick is made of, what it does for your body is a very important thing to know. Even though you might not be thinking about what you are eating while you are eating it, you just want to find something you like. If it tastes good, you're gonna eat it. You really need to know what's in it. So let's watch this short video and then we can talk about it after.
I'm not gonna lie, this is very interesting. I'm sure a lot of you watching this video have heard about this brand right here, Drumstick. You probably have actually eaten these before. You could find these ice creams at like any grocery store. Well, it has recently become a trend to leave these drumsticks out in the open to see if they could melt. I know what a lot of you might be thinking. Well, obviously they're going to be melting because they're ice cream. This trend is pointless. Well, that's where you would be wrong because people are leaving the drumstick ice creams out for 24 hours and they are literally not melting, as you could see right here. This has caused a lot of people across the world to be very confused and concerned because if the drumstick ice cream is actually ice cream, it would melt, but it doesn't melt. So What is it?
What is it? I would love for you guys to really just think about that. Doesn't ice cream melt? I don't know, but I just feel like ice cream is supposed to melt. What could they have possibly put inside this that makes it one so addictive to us? I don't know if there's anyone who doesn't like it, but I like it. What makes it so addictive, and also, what's a part of it? What is it doing to our body? Is that how a food test comes in? Do you really know? Are food tests even important? And why do we even need to test food? Why are we testing food? Let me just give you a minute to think about it, and then we can just have a little discussion in our chat or in our groups, and then we can share afterwards. Now that we have thought about it, are food tests even important, and why do we need to test food? Now, food tests are, of course, very important. I say, kudos to the scientists or the scientists, plural, who came up with these food tests. Now, without the food tests, we wouldn't even have known what particular nutrients our bodies have been absorbing through eating certain foods. Of course, it's very important to know what we're putting into our bodies. And I know I probably am not the only one, but you have persons when you go to the supermarket, you stop, you don't just take up a snack, but you of course look at the packaging and you read the nutritional facts. So of course, before the food is given to you in these packages, certain tests would be run and certain nutrients would be fortified into the food in order to come up with the nutritional facts that we have seen on the packaging. Now, we're reading and we're reading for ingredients, we're reading for the facts, and we just want to know what we are putting into our bodies. They are very important, and we, of course, need to be able to say, well, I am consuming this, I am consuming that. Because really and truly, if we did not know what nutrient was in our food when we consumed it, we'd be basically lacking a particular nutrient. You don't want that to happen. There are varying detrimental effects that can occur as a result of us lacking nutrients. And we just want to know, what we're consuming, how we are making our bodies healthy or not making it healthy. So that's where this Gizmos food test simulation comes in. We are basically going to be using this simulation to test varying food samples, as you can see here, all the way from A straight down to S. They are basically just samples of varying foods that we have taken and put into a test tube in order to find out what nutrients is in there. We're going to be having to conduct these varying food tests. So we have the Benedict solution, the Lugal, the Burette, and the Red Sedan. We're going to be using those to test whether the food have carbohydrates, proteins, and lipids in them. Now, why are we using this simulation as opposed to really just doing it among us here in our physical setting? Well, the thing is, to be honest, the resources are limited, and it's also less likely for you to really make a mistake. Let's say you're conducting this test on your own. You make a mistake, the correct result doesn't come out, you really don't learn anything. Also, we might be playing with fire here with our Benedict's test, and we don't want any of you to get hurt. So we're just going to be doing this online simulation. We also don't really have enough of each of these to really conduct the experiments as really how we want to. So we're going to be experimenting here with this simulation on Gizmos. All right, so let's really just dive into how we're going to be using it. Now, the question is, or the question that, will drive our motivation for this whole entire class is, how do you test for carbohydrates, proteins, and lipids? Now, we're going to be adding a particular food sample from our samples given into the food holder, and then we're going to be just testing them. So we're going to put it in the food sample under Benedict's test, and under all of them we're going to click test. Now the Lugal test, it uses iodine to test for starch, which of course we should know is a polysaccharide or a complex carbohydrate. The burette test uses a solution of potassium hydroxide and the copper sulfate to test for protein, and the red Sudan test uses a fat soluble dye called red Sudan, or sorry, Sudan red, to indicate the presence of lipids. And of course, afterwards, you're going to be running through to see what nutrients sample A contains, of course, when the tests are either going to be negative or positive. All right, so we are going to be using sample D. Now, as I said earlier, you're going to place sample D into our food sample holder and under each test here. So, firstly, we have the Benedict's test. We're going to press test. Now, as test is pressed here, you're going to see that the dropper is used to add some food sample to the test tube, after which we add the Benedict solution, and then it is boiled up until 5 minutes. And if we're seeing a change, which there is none, but we're gonna get into that just soon, we're gonna press test on the Lugal now, and please make sure that you are writing down these results. If there is a change, if there is no change, and we say change, we mean color change. We're gonna do it for the protein now. So the burette solution is gonna be used here. So you add sample D to the test tube, and the burette solution is added afterwards. And there was a change. It's a purple color. And here we are with the red Sudan test. And we add this food sample and then the Sudan red. And then look, there are some changes that we're observing. So, how do we really make sense of what just happened? So, let us just go into the theory of all. Carbohydrates are mainly used for energy by our bodies. So it's one of the main sources of energy. When we eat food rich in carbohydrates, we get a lot of energy to do a lot of things. Energy is basically us just doing work. Anything that involves us doing work is energy. We need to consume foods rich in carbohydrates in order for us to do this, to move, to grow, all of those things. Now, carbohydrates can be classified into four categories: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Two tests now detect the presence of carbohydrates. There's the Benedict's test, as we saw, and there's also the Lugal or the iodine solution test. Now, with the Benedict's test, I'm sure we saw that it was heated. Now, if this was conducted in a real setting, you would have seen the live color changes, but that is not, unfortunately, what we saw there. We just saw one color change. Now, a Benedict's solution turns from blue to green when heated. So it turns from blue to green to yellow to orange to red in the presence of monosaccharides and some disaccharides. Now, if disaccharides or an oligosaccharide is heated with acid, it will break down into its component monosaccharide molecules, and it can be detected by a Benedict's solution. So when we know that food contains carbohydrates here with the Benedict solution is when there is a red color change in the final result. Now for Lugal iodine solution, there is going to be a change from brown to dark purple. So your result should be dark purple if you are seeing any iodine present in your food sample. That is your food sample. Now, or okay, your food as well. Now, here it is. You're gonna see a purple or a bluish purple color being present in the test tube if there is any iodine. Now, proteins have many functions in our body. Hemoglobin in red blood cells carry oxygen throughout our body. Actin and myosin form the cytoskeleton that helps give cells their shape. Proteins are also primary components of our hair, skin, cartilage, and other structures in the body. Enzymes are proteins that catalyze and regulate chemical reactions. And proteins are also the main components of muscle fibers. And of course, as we discussed earlier, fish, egg, cheese, dairy products, beans, peas, walnuts, all these things contain protein. So when you eat all these foods, You're basically absorbing protein through the process of digestion. Not only protein, but this is the focus right now. The burette test, as we saw earlier with the burette solution, is what is used to test or detect the peptide bonds that hold amino acids together. So it's what is used to detect protein. In the presence of proteins, copper hydroxide and sulfate, copper sulfate, turns from blue to purple. So the final result is going to be purple if there is any protein in the sample. Now, lipids are a large group or a broad group of compounds that include fats, oils, waxes, cholesterols, and others. The building blocks of lipids, or of fats, are fatty acids. Now, lipids are found in cell membranes. That's the myelin that insulates nerve cells and hormones such as steroids. And our subcutaneous fat helps our bodies to retain heat. So I see some persons really are going to be way hotter than others based on what I'm seeing here. The fat that is attached to our bodies will determine how hot we are. So it retains heat and it also provides a rich source of energy. That is, of course, when our carbohydrate has run out, we turn over to fat and depend on that for energy. Now, several tests can be used to detect lipids. Now, we have, of course, this test. So, when you buy a bag of chips, of course, you're going to see some transparency on the paper. That is because your french fries, the chips, or the donuts in your bag, it has a bit of lipids in there. Now, of course, you're gonna say, Miss, what if it is that your paper bag got wet with, let's say, water? Now, it's gonna look a bit different because say it got wet with water, it can of course dry. The difference is that lipids, it cannot dry, it will just have that transparent look. Now, if a lipid solution is mixed with ethanol and water, the solution is going to turn cloudy and opaque. But we're not going to be doing that test. Of course, as you saw, we're doing the red Sudan test. Now, Sudan red is a lipid soluble dye. When added to a solution containing lipids, the dye will absorb into the lipids, resulting in concentrated spots of red color. So the solution will be red, but you're going to see little spots within your solution. All right, let's pause here for a second and I want you to answer the questions prior to now. Okay, so the questions dictate, was there any carbohydrate through Benedict's solution test? Was there any carbohydrate through the Lugal test? Was there any protein through the Burette test? Was there any fat through the Sudan red test? And just tell me, based on these answers to these questions, what nutrients does sample A contain? I'm so sorry, we use sample D. What nutrients does sample D contain? The nutrients present in sample D is, of course, protein and fats, and that's just from the food test that we have conducted. So the Benedict test, it really produced no color change after that happened. The Lugal test, as you can see, it is still the same color. There was no change after that happened. But the protein test through the Burette solution, though, there was a color change of purple. And for the Sudan red, I must apologize for saying red Sudan if I have. That's a slip of the tongue. For the Sudan red test, there is, of course, a red color change here with concentrated spots of the lipid. So now that we know that the food contains protein and fat, let's just look at our list here. We want to find out what kind of food is sample D most likely to be. Is it going to be apple juice? Does apple juice contain protein and fat? Is it gonna be baked beans? Does baked beans contain protein and fat? Is it gonna be oatmeal? Does oatmeal contain protein and fat? Or is it going to be scrambled eggs? Does scrambled eggs contain protein and fat? Now, giving you a minute to think about that. If you said scrambled eggs, then you are correct. So scrambled eggs contain protein and it also contains fat. That is most likely what they would have used to prepare, that sample found in test tube D there. So, what I want you to do now after that demonstration is you are going to be answering some questions through your own exploration of. Okay, so this is the activity that I'm going to give you 10 to 15 minutes just complete amongst yourself in your groups. The question is, what nutrients does each food sample contain? Now, you are going to use the four available tests in our simulation to find the nutritional content of each samples E, G, and M. Now, sample A has been done for you as an example. And of course, you're going to record the results on the table below. So if you test sample E and you realize that the Benedict test is positive, you put a plus. If you realize that it's negative, you're going to put that little subtraction symbol. On the other side, test results. Are these nutrients present? Now, if you put a plus, you're going to put yes for your answer. If you put the subtraction sign, you're going to put no for your answer. Now, just to remind you how you are going to use this simulation to conduct this test, this is what you're going to do. Now, we would have just done this test with sample D. Now, if you had continued and you had done it while I was doing it, you can press reset and you are going to now search for E, drag it to the food sample holder, and you're going to press test under each test. Now, of course, when you come up with the results, you should already know what's going to happen. Now, for the Benedict test, you should know the change that will happen. For the Lugal or the iodine test, you should know. For the Burette test, you should know. And remember, for the red sedan test, you should also know. Okay, so now that our activity is completed, these are the results you should have come up with. So sample E only contains proteins and lipids. Sample G really contains none of the above nutrients. Thank you for watching this lesson. However, this is only a snippet of a lesson that was meant to be 1 hour long.